Howdy, my name is Stephanie Aranda and I will be presenting the foot and mouth disease fmdv. This subject is covered in Chapter 11, Family Picoronaviridae in Susan Payne's book Viruses from Understanding to Investigation. The first thing that you guys should not be confused about this disease is that it is not hand, foot, and mouth disease, fmdv. DV, yeah. This disease does not affect cattle, but it is most prevalent in infants. The foot and mouth disease, which is a different pre-coronavirus, is the main topic of this presentation that affects cattle, sheep, pigs, and horses. The family of the foot and mouth disease is from the Picoronaviridae family and has the aphthovirus genus. Affecting cattle, sheep, pigs, and horses, this disease is a vascular disease that is easily transmitted from long distances in the air to contact. An example would be either direct contact or through fomites. This Picoronaviridae is undeveloped virus, another word for a naked virus, that is only composed of the genome and protein capsid. The viral genome is a positive sense and is a single-stranded RNA that ranges in size of approximately 7,500 to 9,000 nucleotides NC, with the acosahedral symmetry T equals 3, which refers that the capsids are built from 180 capsid proteins. The viral replication cycle of this pre-coronavirus consists of 5 steps, with the undeveloped virion particle infecting its host cell. Steps 1 and 2 being the virion's attachment and penetration to allow the release of the RNA into the host cell. Since the virion replication is cytoplasmic, step 3 allows for viral RNA replication and translation to progress within the cytoplasm. In step 4, the capsid proteins are translated and assembled within the cytoplasm prior to the release of the virus particles in step 5, prior to the maturation of the virions outside of the host cell. This positive sense viral RNA information that has been established from the 4th slide would result in the immediate translation by the host cell, unlike the negative sense viral RNA would. Due to the virus replication being located in the cytoplasm, genome replication requires the utilization of the cell membrane complexes from the host cell. The genome is attached covalently to the 5' end of the mRNA seen in this figure. The primer used for this genome synthesis is the protein primer VPG. This is a short polypeptide that is linked to the 5' ends of the newly synthesized viral RNAs. For genome replication to take place, the ribosomes must be freed. Then the VPG protein primer is then used to initiate the RNA synthesis. The synthesis is heavily favors the genome strands. Thus, the cRNA is the template. The formation is efficient on the 3' end of the cRNA. Okay, for transmission, I will be discussing the transmission rates worldwide in this slide. This diagram was obtained from the Frontiers Organization website as of 2013. As we can see that the foot and mouth disease can be found worldwide. This vascular disease can be found affecting mainly the undeveloped parts of the world as an endemic. Countries that are free of this disease often take many precautions to retain the title of being FMDV free, which includes the United States. These precautions can include, but are not limited to, vaccinations and surveillance of the livestock for any clinical symptoms. With this, there is hope to prevent any outbreaks in these FMDV-free countries, which, if it were to happen, would result in huge economic losses and destruction of many animals who are either contacted or infected or in close parameters. As you can see here, the estimated minimum doses for injection vary between species and display the most prevalent susceptible hosts, such as the pig. Okay, for pathogenesis, the development of this disease could result from entryways on the mouth, nostrils, and feet. In this figure, we can see how the spread of the virus could affect the entire host in a matter of 7 days throughout the body, causing lesions in the listed areas such as the tongue, gums, bulb of heels, and other body parts. Disease manifestation. The physical characteristics caused by the foot-mouth disease virus are vesicles in the mouth, tongue, hoofs, and nipples. There is no found treatment for this easily transmissible disease, as it is handled by the destruction of the infected animals or prevention through the inactivated vaccines that are produced in the worldwide expensive biocontainment facilities, which they use the virulent strains of the foot and mouth disease. Okay, for diagnosis, besides the physical characteristics introduced by the previous slide, other characteristics that are also monitored is the increase in body temperature and appetite loss. To confirm the diagnosis with the visual evidence from the livestock animal, other tests could be done to have direct confirmation. This could include live virus inoculation, which is used to detect viral components from the affected animal. As seen on the diagram, there are various tests that are used to identify specific viral components, such as utilizing the RT-PCR, the reverse transcription polymerase chain reaction, to detect the viral nucleic acid by utilizing the DNA marker, as seen on the figure. These are the resources I have utilized in preparing this presentation. I have used Susan Payne's book, Chapter 11, Family Picoronaviridae, as my most used resource when collecting information. And this concludes my presentation on the foot and mouth disease. Thank you very much and have a great day.